Okay, thanks, Joe. So I'm going to give an update on pneumococcal vaccines. The title says I'm going to talk about protein vaccines, which I certainly will discuss, but I'm also going to spend some of my time talking about pneumococcal conjugate vaccines because I think some of the major developments in the field in the last 12 months or so have been in the areas of conjugate vaccines. We heard this morning about the Merck 15-valent conjugate vaccine that's in phase 2 clinical trials, and there are a number of other developing country manufacturers that have a lot of interest in conjugate vaccines. Plus, there's been the introduction of pneumococcal conjugate vaccines in a number of developing countries over the last year or so. So this is just a reminder about the global burden of disease. Pneumonia is the #1 killer of children under the age of 5. This is the most recent data. So pneumonia is responsible for more deaths in children under 5 than HIV, malaria, and measles combined. And the major cause of pneumonia, severe pneumonia, is Streptococcus pneumoniae. This next slide shows you where most of the deaths are occurring in these children, and it's pretty obvious to see where this is happening. It's primarily in Africa and parts of Asia, particularly India, Afghanistan, and Pakistan. A little bit more about the organism. So as I mentioned, Streptococcus pneumoniae is the leading cause of severe pneumonia. It is a complex bacterium that has more than 90 serotypes, so even with stepping up to a 15-valent conjugate vaccine, which does cover the majority of the disease-causing serotypes, there's still a gap in protection coverage. It is a cause of other life-threatening diseases, and this includes meningitis and sepsis. The organism is also a major cause of otitis media. And those with weakened immune responses are particularly susceptible to pneumococcal infection. So most disease occurs in very young children under the age of 2 or in the elderly. And certainly there has been emerging antibiotic resistance amongst pneumococcal types causing disease. So this is a list of the currently licensed pneumococcal vaccines. Prevnar 7 from Wyeth was licensed in the US in the year 2000 and has been used very broadly throughout the world since that time, and is gradually being replaced by the new version, Prevnar 13. The 7 serotypes in Prevnar 7 were really selected based upon their prevalence of causing invasive disease in North America and Europe, and did not include some of the serotypes that are actually causing a lot of the disease in the developing world, and this includes serotypes 1 and 5. So the newer pneumococcal vaccine, conjugate vaccines, Prevnar 13 as well as Synflorix 10, do include those particular serotypes 1 and 5, and some other serotypes as well. So we're up to 13 now, or as we heard from the Merck vaccine, it's the same 13 in Prevnar plus 2 additional serotypes. So what's been the impact of introduction of the vaccine? This is just some data from the US showing the impact of PCV7, Prevnar 7, over a period of time. And what can be seen is, on the very left-hand side, is the pre-vaccine introduction data. And if you look at the dark part, the dark bars, that's the disease caused by the serotypes included in the vaccine. And so after introduction of the vaccine over time, there's been a gradual and dramatic reduction in disease caused by serotypes included in the vaccine. However, there's also been a concomitant increase in disease caused by serotypes not included in the vaccine. And that's shown by the top part of the bar, so the kind of the light blue coloring. So this is presumably due to either serotype emergence or serotype replacement. And so the overall net benefit of the vaccine certainly is positive, but it's been somewhat dampened by the emergence of these non-vaccine serotypes. So with the introduction of the higher valency vaccines, some of this will get covered, and so it's certainly going to be interesting to keep monitoring this, what will happen with the 13-valent vaccine. It's also going to be of great interest to see what these vaccines are going to do as they become introduced into developing countries where the disease burden is much higher than it is in the US. So just a quick summary on the conjugate vaccines and what we know about them. So clearly they have proven efficacy and effectiveness. There's high efficacy against the serotypes that are actually included in the vaccine. There's no question about that. In a number of studies, there have been shown to be an impact on pneumonia, 20 to 35% reduction in radiological pneumonia. So this is all-cause pneumonia. So Streptococcus pneumonia is thought to be responsible for about 40% of the cases of severe pneumonia. I think importantly from the path standpoint, since our focus is on developing vaccines for children in developing countries, there have been some clinical studies in Africa, in the Gambia and in South Africa, that have shown a 9-valent vaccine that was under development at Wyeth and has now been replaced by the 13-valent, that this vaccine did have an impact on invasive disease and pneumonia, and actually in the Gambia it had an impact on all-cause mortality. So these vaccines do work in developing countries. A big plus for the conjugate vaccines is the regulatory path. There are WHO guidelines now for approval of vaccines based upon non-inferiority to existing licensed vaccines. So it's based upon immunological readouts, not efficacy studies. So for emerging market manufacturers, this is a big plus that streamlines the development of these vaccines without having to do large-scale efficacy studies. And then finally, an important factor about the conjugate vaccines is their ability to promote herd immunity. So by vaccinating young children who now do not transmit the organism as much as they would have, the indirect effects of this vaccine are actually twice what they are on the children that actually receive the vaccine. I want to touch on the pneumococcal pilot advance market commitment, which was mentioned very briefly this morning. This was an incentive put together to try and get vaccine manufacturers, really it has two goals. One is to encourage the existing manufacturers with licensed vaccines to expand capacity of their vaccines and to supply the vaccine through procurement agencies such as Gavi to developing countries. And secondly is to encourage new vaccine manufacturers, in particular developing country manufacturers, to enter the market. There's $1.5 billion put up for the advance market commitment. As I mentioned, it's to try and create incentives for producers, and the long-term goal is to catalyze competition to drive down the prices of the vaccine over the long term. So the result is that the vaccine is now becoming available to developing countries much quicker than it probably would have been without the advance market commitment incentive being in place. So currently, right now, the agreements that Wyeth and GSK have is to supply the vaccine initially at $7 a dose. $3.50 will come from the AMC, and $3.50 comes from Gavi and the country that's actually requesting the vaccine. And the long-term tail price for this vaccine will be $3.50 a dose. So some of this coming from Gavi, some from the country itself. But these vaccines are now starting to be introduced into developing countries, and I'll describe that more in just a moment. The funding is primarily from the Bill and Melinda Gates Foundation and the countries of Canada, Norway, Russia, and the United Kingdom. And as I mentioned, the vaccine manufacturers that are currently participating in the AMC, that actually have contracts under the AMC, are Wyeth and GSK, although there are other manufacturers who have signed up for interest in this, no contracts at this point in time. But these companies, which include some manufacturers in India, hope to be able to supply vaccines through the AMC in the future. So this is kind of where we were about a year ago on introduction of PCVs in developing countries, or at least in Gavi-eligible countries. So there were two countries that introduced the vaccine in 2009, and this is the Gambia and Rwanda. This was actually through a donation from Wyeth together with the Gavi Alliance. But since then, in the last year or so, a number of other countries have started to introduce the vaccine, and these are listed here. And so you can see some in Central South America as well as African countries, and it's thought that by the end of this year that there will be 19 developing countries that will have actually introduced pneumococcal conjugate vaccines through the AMC mechanism. To switch now to what we're doing at PATH, this is actually our pneumococcal vaccine development portfolio, and we have a combination of PCVs and protein vaccines, and really it's based upon kind of timelines. The short-term strategy is to try and encourage new manufacturers to come up with cheaper PCVs, and the longer-term strategy is to try and develop protein vaccines, or maybe protein vaccines in combination with PCVs. So we have a number of partnerships on the common protein vaccine front. They're listed here. So first, Intercell, we've had a long-standing collaboration since 2006 with Intercell. Recently completed a phase 1 trial with their recombinant protein vaccine, which showed that it was safe and immunogenic. We have a number of preclinical programs, research programs with the University of Glasgow, Genocea Children's Hospital Boston, that I'll tell you a little bit more about in a moment. Mucosis, another one of our partners that's present and will be talking at this meeting later on. I know there's going to be a presentation from Genocea about their HSV program later today. And also an academic consortium of University of Adelaide, University of Alabama, and St. Jude's Hospital that has a candidate in preclinical development. Another program that we have is on a killed whole cell vaccine, and this is with Children's Hospital Boston in partnership with the Butantan Institute in Brazil, and I'll give a very brief update on that project. That's targeted to enter phase 1 clinical trials by the end of this year. We have a partnership with GlaxoSmithKline and the London School of Hygiene and Tropical Medicine and the Medical Research Council of the Gambia to run a phase 2 clinical trial of GSK's protein plus conjugate vaccine in children in the Gambia, and that study began earlier this year. It has a toddler cohort that's completed enrollment and has completed all injections, and then we'll be moving on to a study in infants in the not too distant future. And then finally, our conjugate vaccine partners. So we have a partnership with the Chengdu Institute of Biological Products based in China and with the Serum Institute of India with a PCV vaccine that is targeted to hit the clinic towards the end of this year. So as I mentioned, the currently licensed vaccines are starting to get out there and are saving lives, but there are a number of shortcomings of these vaccines. One of these, even with the assistance, these vaccines from Gavi and from the AMC are still not cheap. And they're certainly unaffordable without assistance from organizations such as Gavi. So one of our goals is to try and get the inherent costs of the vaccines down by trying to streamline the manufacturing process and work with developing country manufacturers. They also only cover, as I mentioned, some of the 90 plus serotypes. That's where the protein vaccines come in, is to try and give that broad coverage. And these vaccines aren't typically tailored to the needs of developing countries. So as I mentioned with Prevnar 7, those initial 7 serotypes were based upon prevalence in North America and Europe, and not necessarily the most prevalent serotypes in the developing world. So our strategy for developing lower cost conjugate vaccines is to try and develop technologies that will improve the efficiency. So we did an analysis of the cost drivers for making pneumococcal conjugate vaccines, and it's really based upon the prime cost drivers are the protein carrier, the production of the polysaccharides, and then the conjugation chemistry itself. And so we've been working with ways to try and improve the efficiencies of all 3 of these components. The target is to provide coverage against the serotypes that are most prevalent in the developing world, and I'll give an example of that in just a moment. And by doing this, the ultimate goal is to reduce the cost of these vaccines. And one thing I do want to mention briefly is what we think may be a next generation strategy is to incorporate a common protein as the carrier for a pneumococcal conjugate vaccine. The idea behind this is that we'll still follow the regulatory path of a conjugate, traditional conjugate vaccine, but may have additional coverage by using a pneumococcal protein as the carrier, which could be demonstrated in post-licensing studies. So this is the serotype coverage that we're looking at. On the left, we've got Prevnar 7, and as you can see, if we're looking just at, this is just looking at Africa and Asia, and you can see the coverage is around 50%. And that's because serotypes 1 and 5, which are quite prevalent in the developing world, are not included in that vaccine. If you actually take the 7 or 8 most common serotypes in the developing world, you can get vaccines that clearly meet the requirements of the advanced market commitment, which are 60% coverage of, at least 60% coverage of the serotypes in the regions of interest. And by going up to the most common, 8 most common serotypes, we can get basically the same coverage as you can see for Synflorix, which has 10 serotypes included, and not far off what you see with Prevnar 13. Just briefly, the strategy I mentioned about incorporating a common protein, one of the prime targets we believe that could be used as a carrier protein is a protein called Pneumolysin, or the toxoided version Pneumolysoid. This is an interesting protein because it has some interesting biological properties. First of all, it's a toxin, so there is the possibility of generating correlate of protection using toxin-neutralizing antibodies as a readout, and this is not always the case for other protein vaccines. So it also is involved in complement activation and has been described to have inherent adjuvant properties, which make it a particularly appealing protein to take a look at. There are a number of candidates out there that we've been looking at, one from the University of Glasgow and Tim Mitchell, Rod Tweeton at the University of Oklahoma, and then James Payton at the University of Adelaide has several candidates we've been taking a look at. So I've touched on most of these points, but let me quickly run through them again. The shortcomings of the pneumococcal conjugate vaccines and why we're particularly interested in protein vaccines. One is the manufacturing complexity. When you're up to a 13 or 15 valent vaccine and you have to produce 15 polysaccharides and QC released 15 polysaccharides plus do the conjugation on all 15 conjugates, and then again the release assays on this, it adds up to hundreds and hundreds of different assays. It is probably the most complex vaccine that's currently being produced. As a result of that complexity, these vaccines are relatively expensive to make. We touched on the serotype-specific coverage and the serotype replacement or emergent issues. There's some suggestion that efficacy of some of these serotypes may be limited, particularly serotypes 1 and 3, and evidence that effectiveness against mucosal disease is not ideal compared to invasive disease. So the reasons for looking at protein vaccines is the potential for breadth of coverage against all serotypes. So we look for proteins that are well conserved against all strains, all serotypes. By incorporating 2 or maybe 3 recombinant proteins, it's a less complex manufacturing process and therefore should be less expensive. And by targeting proteins that are involved in different parts of the pathogenesis of the organism, there's the chance to get broad coverage against all aspects of disease, such as nasopharyngeal carriage, pneumonia, lung colonization, and then invasive disease. Just want to touch on a couple of our partnerships in the area of protein vaccines. One that has moved well in the last year or so is a collaboration with Children's Hospital Boston with Rick Malley's group on an inactivated whole cell vaccine, and this is a partnership that we have also with the Butantan Institute in Brazil. So this is a non-encapsulated strain that is autolysin deficient and has had its pneumolysin gene removed and replaced with a pneumolysoid. Interesting thing about this vaccine is that it has been shown to have multiple mechanisms of protection. So it's both, protection is both T-cell and antibody dependent. And I'll show you just a little bit of data on that. So this is in preclinical mouse studies looking at nasopharyngeal carriage. And on the left-hand side, we have our negative control. So this is the adjuvant alone. So this is given subcutaneously, the whole cell formulated in alum. And if we increase if we add 10 micrograms of the vaccine in the middle, we see about a 1 log reduction in CFU in the nasopharynx, and then at the higher dose, there's about a 2 log reduction. And this has been shown by Rick's group to be dependent upon both CD4 positive T cells. So if you deplete CD4 T cells, this protection goes away. Or if you deplete IL-17A, the protection also goes away. So this, the mechanism of protection against carriage is thought to be mediated by Th17 T cells. This is a different model. It's a sepsis model where the bacteria are actually administered intranasally but end up causing a lethal sepsis. And in this particular model, most of the animals die between 4 and 5 days post-challenge. If they've been vaccinated with the lower dose of the vaccine, there's about a 70% protection, and then 100% survival if the mice receive the higher dose, 100 micrograms of the whole cell vaccine. And this particular model, the invasive disease, is thought to be mediated by the antibodies. So you can actually you can passively transfer serum from immunized animals and show that it protects in this model. So the hope for this vaccine is that because it's a complex mix of proteins, it can elicit both T cell and B cell immunity, and it should be able to be produced at a very low cost. This may be a potential solution for preventing pneumococcal disease in developing countries. As a spin-out to this project, we'd been working with Rick Malley at Children's Hospital for a number of years, but we really had no idea what was the target of the T cells involved in protection against the nasopharyngeal carriage. Independently, we were approached by a company called Genocea, coming from the Cambridge area, who had approached PATH with a technology for discovery of T cell antigens using basically an expression cloning strategy. And so we basically put these two groups together. So it turned out that Rick Malley at Children's Hospital knew one of the scientific founder at Genocea, Darren Higgins, who's at Harvard. And these two groups got together and pitched a project to us, and we funded it to try and identify the antigens being recognized by those protective T cells in Rick's model. So basically, Genocea made up the library, the pneumococcal library, together with Children's Hospital. They screened the library and came up with a number of candidates. So in the mouse system, there were 42 antigens identified, and then these were plugged back into the model to see which ones were being protective. This has also been done with human Th17 cells as well at Genocea. And I know, as I mentioned, there's going to be a talk later on today that will describe the Genocea technology in more detail. So I'm not going to go through the Genocea technology in this presentation, since we're going to hear more about it later today. But just to say that we have identified a number of candidates using this, and they are being, or have been, or are being screened for their ability to now protect back in the model that was used to identify them. So this is some of the data that's been generated taking some of these candidates that were identified in the mouse system and putting them back into the model. And if you look at the left-hand side, I think I can use a pointer. We have the very left is the whole cell antigen, which is our positive control. The next one along, ICP47, is actually a control protein that comes from, it's an HSV recombinant protein expressed in E. coli. So this is a negative control. And then we have 3 antigens that were identified through the screening process at Genocea and now show good protection in this model. So SP2108, SP0148, and SP0882. The next one after that is one that was identified in the screen but did not turn out to be protective upon reimmunization. And then the final antigen on the end there is actually an antigen that was identified as a B cell target, not as a T cell target. And I just will wrap it up here. To finalize, what I wanted to mention, which I didn't mention earlier, is that PATH is all about partnerships. We actually, on the Pneumococcal Vaccine Project, we don't actually have any labs working directly on this. We do everything through partnerships. So we partner with academic organizations. I mentioned some of them today. We partner with biotech companies. Again, I mentioned some of those today, as well as big pharma and other global health organizations. I think the ones being circled now are the ones that we actually have vaccine development partnerships with. And I'll leave it there. Thank you very much.
Do we have a microphone?
Hey, Mark. That was a very nice talk. Thank you. You mentioned that the current vaccines are not ideal for getting into these Gavi-type countries. The question I have is, are you putting any pressure on the manufacturers or this early research for looking at vaccines that are more thermal stable than what's available nowadays?
I caught part of that. I got the last bit about the thermostable. I didn't get the first part.
So the question is, when you're developing these early stage, in fact with Genocea, the leads and things,
yeah.
Are you screening for things that are more thermal stable that would be easier to distribute in these countries?
Yeah, I mean that's a longer term objective, certainly, and as you may be aware, PATH actually has a technology solutions group that's working on stabilization of vaccines. And the challenge is when you bring in the technology, because right now, the rush right now is to try and get a low-cost conjugate, so in the short term, a low-cost conjugate vaccine as quick as we can. And that's really driven by the shortfall in funding at Gavi and the cost of these vaccines. So Gavi having to pay, so forget the AMC, Gavi's paying $350 or close to $350 a dose for pneumococcal vaccines, and that adds up with estimated demand, peak demand of over 200 million doses a year. That's a huge amount of money. So the real push, short-term push right now is to get lower cost conjugate vaccines out as quick as we can. And so to do that, there's not going to be work done on stabilization, but in parallel, I think in the longer term, clearly that becomes an issue. And so the longer term strategy, protein vaccines, when we actually get something that's starting to look good, those are issues we'd certainly like to look at. But the challenge really is when you do it, and not doing it too late, because as I'm sure you're aware, once the vaccine gets licensed now, to have to go back and redo those studies and redo clinical trials, the challenge is when's the most appropriate time to try and bring those technologies in. So for the conjugate vaccine, at least the ones we're really pushing hard now, we're not trying to do this, but certainly we will in the long term.